Hello, welcome to Lekpa tutorial. In today's tutorial, we'll be looking at cells of the nervous system, particularly the neurons and the neurog. My name is Dr. Lekpa David. I hope you will enjoy today's tutorial. So let's look at the neurons. The neuron is a functional unit of the nervous system. Without the neuron, the nervous system cannot function effectively. Without the neurons, there won't be any transmission of impulses within the nervous system. So the understanding of the neuron is key in understanding the nervous system. There are millions of neurons in the body connected together to ensure that there's effective transmission of impulses. Now, these neurons can be classified into various types. It can be classified anatomically, based on the anatomical structure, into unipolar, bipolar, and multipolar neurons. A unipolar neuron is a type of neuron that has just one particular process, whereas a bipolar neuron is a neuron that has two processes, and the multipolar neurons are neurons that has several processes. I will explain what I mean by that as we continue in the tutorial. Now, neurons can also be classified physiologically based on their function. So neurons can be classified based on their structure, which is anatomically, and based on their function, which is physiologically. So physiologically, neuron can be classified into sensory neuron. Sensory neuron, the neuron that convey information, impulses from the external environment through the spatial senses to the brain for interpretation. Then we have interneurons. Interneurons are neurons that is between the sensory and the motor neuron. And the motor neuron is the one that conveys the already interpreted information back to the organs, the effector organs. So those are the three types of neuron. I take it again. So anatomically, a neuron can be classified into unipolar, bipolar, and multipolar. Unipolar meaning it has just one process. When I explain a typical neuron to you, you understand what I mean by the process. Then bipolar is the one that has two processes, and multipolar is the one that has multiple, from the word multiple, several several processes. Now, physiologically, neurons are classified into three as well: sensory neuron, interneurons, and motor neurons. Sensory neuron conveys sensory information. Interneurons, inter meaning it connects both the sensory and the motor together, and then a motor neuron that conveys information from the brain to the spinal cord and to the effector organs. Now, between these multiple millions of neurons that are found in the nervous system, when neurons come together, they form what we call synapse. Synapse. The synapse is the functional relay point between two neurons or between neurons and effector organ. So this synapse, your synaptic cleft, is a synaptic space, is a place where a lot of transmission of information, which I'll also explain later, occur. So there's a synapse, a space, a potential point between neurons. So that's how neurons connect. Between one neuron connect, there's a synapse, another neuron, there's a synapse, like that, and the entire connections. So we also have neuromuscular junctions and neuroglandular junctions that are found within the nervous system. Now, let's go straight to parts of the nervous system. We need to know the various parts of the nervous system. The nervous system is made up of two particular parts. We have the cell body, and then we have the processes. We have the cell body and the processes. The cell body, which is the soma or pericardium, is made up of a lot of nucleus, cytoplasm, and the various organelles. Just as we have organelles in the cell, we have all those organelles that are found in the neurons. So like the Golgi apparatus, nucleus, mitochondria, and the rest of them. Then we have the processes. The processes is made up of the dendrites and the axons. The dendrites and the axons. The dendrites are highly branched processes that are coming out of the cell body. They are numerous, they are branched like a tree, and they are very short, usually short. And they convey afferent function, afferent information to the neuron. Now, the axon is a cylindrical process, just singular cylindrical process, long and thin, that is usually myelinated, although there are some unmyelinated neurons, and this has to do with efferent function. So those are the two processes, the dendrites and axon. So the neurons are made up of two components, cell body and processes. Cell body consists of the nucleus, cytoplasm, and other cytoplasmic organelles. Then the process is made up of the dendrites and the axons. Now, this is a typical neuron. Typical neuron. So, as I've said earlier, this is the cell body. The cell body. You can see this rounded structure here is the cell body. So we have the cell organelle, the nucleus, and the rest structures that are found, the neurofibrin, the missile body. The missile body is a special organ found in the neuron that is not in the normal cell. Not in the normal cell. So this, then after the cell body, you can see three light structures coming out of the cell body. These are called dendrites. They are short branch processes. Short branch processes. They are called the dendrites. They are called the dendrite. Now, after this cell body here, we have the helix or the neck of the neurons where action potential are generated before we now have the entire axons. The entire axons. This is the axons. This is the axons where synaptic information or impulses are being carried. So we have the cell body, the dendrite, the axons, and then toward the end of the axons, we have axonal or synaptic terminal, axonal terminal that contains some synaptic ball. In those synaptic ball is where we have synaptic vesicles. There are numerous vesicles that are found in the synaptic ball. Now, when there is entrance of calcium ion, it causes exocytosis of the synaptic vesicle to bring out its contents, to shrink and then bring out the content of the synaptic vesicle, which is neurotransmitters. So the neurotransmitters are found in the synaptic vesicles within the synaptic ball. Now, these neurotransmitters ensure that there is transmission of these impulses from one neuron to another. And these neurotransmitters can be classified into two: excitatory neurotransmitter, the one that ensures that there is continuous stimulation or transmission of that impulses. And then we have, which example is acetylcholine. Acetylcholine. Then we have the second example, the second type of neurotransmitter, which is inhibitory neurotransmitter, known as GABA. Gamma aminobutyric acid, GABA. Now, if you do histology of the neuron, using Nisi stain, this is what you will see. Nisi stain will ensure that we see the structure of the neuron histologically in an histological state. Now, the second cells that are found in the nervous system are called neuroglia. They are called neuroglia or glia cells. That's supporting cells. They are basically support or nurture as well as protect the neurons, neuroglia. There are types of neuroglia that are found in the brain. 4 types of neuroglia in the brain. We have the astrocytes, we have the oligodendrocytes, we have the microglia, and the epidermal cells. So we look at them one by one. But take note, there are 4 neuroglia: astrocytes, oligodendrocytes, microglia, and epidermal cells. These 4 I have mentioned are found in the central nervous system. Central nervous system. Now, we have another type that is found only in the peripheral nervous system. It is called the neurolimocyte or the Swan cells. It is only found in the peripheral nervous system. So totally, we have 5 types of neuroglia: astrocyte, oligodendrocyte, microglia, epidermal cell, and neurolemocyte, or Swan cells. Now, having known the various types of neuroglia cells, let's look at them one by one. So let's start with astrocytes. Astro, from the word astro, has to do with star. So astrocyte, meaning a cell. Site means cell. So a cell that has a star-like shape, and this astrocyte has many processes. They participate in metabolism of neurotransmitters. There's the neurotransmitter we've talked about that is found in the synaptic vesicles. These astrocytes participate in the metabolism of the neurotransmitters. They help to maintain calcium balance for generation of nerve impulses. Astrocytes also participate in the formation of broad blood-brain barrier as well as brain development. Okay, they are involved in brain development and help in the formation of broad blood-brain barrier, so that substances that are not needed in the blood, in the brain, can be filtered out without them getting into the brain. So these are the functions. And another one is that they provide a link between neurons and blood vessels. So these are the various function of astrocyte. Now, this is the structure of astrocyte. You can see how the astrocyte look like a cell, numerous processes. The second one is oligodendrocyte. Oligodendrocyte are small cells with fewer processes, unlike astrocyte that was having a large, numerous processes. These oligodendrocyte has fewer processes. They form a supporting network around neurons by producing lipids, proteins wrapping around the neuron. And this supporting structure that they form is called myelin sheaths. So they wrap around the neuron to ensure that there is proper transmission of impulses along the axonal pathway. This is you can see the shape of oligodendrocyte. The processes are very few, unlike the astrocyte. Now, the next neuroglia cell is MicroG cells. They are small phagocytic cells that protect the central nervous system by engulfing and invading microbes. So they help in protecting the nervous system when bacteria or foreign bodies enter into the nervous system. They phagocyte, they engulf around the bacteria or the microbes, or the foreign bodies that enter into the brain or the nervous system, and destroy them. So remember, in our introduction, we said that they are supporting cells. So they clear away the debris from dead cells, all the dead cells in the body. So this is the structure of the microglia cells. Now, the next supporting cell is epidermal cells. These are neuroglia cells that line the brain ventricle. In the brain, as we continue this tutorial and another edition, we'll talk about the ventricular system of the brain, where the cerebrospinal fluid is produced. So in the brain, that is where you see the epidermal cells in the colloid plexus. So they help to produce cerebrospinal fluid. So they line the central canal of the spinal cord as well, and help to form and circulate the cerebrospinal fluid within the brain. This is the structure of the epidermal cells. So I've talked about the 4 supporting cells of the central nervous system. Now let's look at the one of the peripheral nervous system. Remember, it's just one, the neurolemocyte, which is Swan cells. These Swan cells produce myelin sheaths that covers the axon in the peripheral nervous system. Remember that the oligodendrocyte form the myelin sheath in the central nervous system. But when it comes to the peripheral nervous system, it is the Swan cells or the neurolemocytes that produces the myelin sheath around the peripheral nervous system. So in conclusion, we've talked about the supporting cells. These supporting cells, they help to protect, to nourish the brain and the entire nervous system to ensure that they function effectively. So I hope you have enjoyed today's tutorial. By the next tutorial, we proceed to deeper structures of the nervous system. Thank you and see you then.